Okay, so we're resuming our AI for Healthcare webinar series. We would take pause for over the summer, but now we're coming back, and this is the first one for this fall. And it's my great pleasure to introduce Professor David Page, James B. Duke Distinguished Professor and the Chair of Biostatistics and Bioinformatics at Duke University. And Dr. Page is a leading researcher at the intersection of machine learning, causal inference, and biomedicine. His pioneering work developed algorithms that makes neural network more interpretable and causally accurate, and with applications spanning electronic health record, genomics, clinical decision support. So today he will share his latest work in brain Causal. I had the privilege to know David for many years, and he's one of the very few individuals I know that who's not only very deeply technical in machine learning statistics, but also actually has profound understanding and interest in clinical tasks and the complexity of EHR data. So without further ado, let's welcome Professor David Page.
Thank you so much for that, Jiming. And it's been an honor for me to know you through the years. It's an honor for me to be here today. I'm a proud Illinois CS graduate, and so I'm delighted to be here with you. Let me just say briefly, when I'm doing this, I have a terrible time seeing the chat. So if anybody has a question, feel free to unmute and ask, or maybe, Jimmy, you can monitor the chat for me.
I'll monitor that and let you know. Yeah.
Great, thank you. And in case I don't leave enough time and run out of time at the end, I want to go ahead and acknowledge a number of collaborators, some who worked with me back when I was at Wisconsin, including people at the Marshfield Clinic, and a number of collaborators here at Duke. And I'll also call their names out as we come to various pieces of work. Some of you have seen a few of these slides at the start before because I think they give some appropriate background, but we'll get to some new technical stuff later in the talk for sure. A couple of years ago, I was giving this talk at ICML and at the big neurology conference and transplant surgery conference, and made the point that I thought what AI was going to really do in clinical trials would be to enrich recruitment for conditions that are either underdiagnosed or that we don't realize people actually have them until much later, including many patients with Alzheimer's, for example. And so you want patients where you can make interventions now that may not really show the big results until 20 years in the future. And so I think AI has a lot of potential there. But also in clinical trials in particular, we've long had hope for reinforcement learning. But of course, the big problem is you don't want the RL algorithm making treatment decisions for patients just so it can learn more. You really want to know that when it's choosing between different options, that those options are in equipoise, and we don't know for sure which of those is better at the moment. And then, of course, everybody's super excited about the large language models, like what's scribing this lecture now, and I'm very excited about that and the potential for trials as well. But of course, it also goes far beyond trials to analysis of observational data, things like adverse drug event discovery and drug repurposing. But this was the kind of message I was giving a couple years ago. And what I said a couple of years ago has only grown more dramatic with last year's Nobel Prizes, for example, in foundation models for health. And of course, the ambient scribes are having a huge impact whenever we go to see our clinicians. Protein language models are having an enormous impact. And when I spoke at an ICML workshop on protein language models, I basically said that my view is I'm not developing the best new protein language models, so I can sit back and watch this change, and if I feel like there's a shortcoming, it's a good bet that in a couple of years that shortcoming is going to be fixed. And so what I really want to focus on, and I put towards, the word towards at the start of my talk, because I'm not claiming to have done this or to be the person that will get there first, but I just want to talk about some contributions towards a foundation model for all of health. Let me come back to this. Let me jump ahead for just a second. What I mean is not only all the literature, but all the EHRs that are distributed and siloed across so many healthcare systems, the wearable data that we'll have to get from patients. That's more patient controlled at this point. My colleague here at Duke, James Jung, works a lot on surgical videos, biological knowledge graphs like we're developing in NHGRI's impact of genetic variants on function program, and a variety of scales of knowledge. So Scott Soderling, who's chair of cell biology here, is doing remarkable things with Rohit Singh around protein language models to actually design their experiments in the lab. Can we put all of this together into a foundation model for health? And I think the key to figuring out how we're going to get there is to look at how we've had the dramatic rapid success in AI of the last decade. And I know all of you know this, but just to recap, we continually come up with new architectures, and I think that needs to continue. Also, just huge amounts of data and processing power. And the self-training trick we see over and over again applied, but really working on many different tasks with the same system, like the early influential papers before the attention is all you need paper. And the few years before that, in NLP, there was a move towards doing more and more with one system until we had one system that you would just put in a natural language prompt. And it could address everything from machine translation to information retrieval to knowledge discovery. And so a variety of NLP tasks could all be addressed with the same system. I've long had an interest, especially in the EHR data, so I want for a little while to talk about just what can we do with EHRs. And it turns out there are many different tasks we can address. So just in that same spirit that we could do knowledge extraction or information retrieval with the same NLP system, can we address all these tasks with the same AI system sitting on top of an EHR? And I would argue that as of right now, today, we cannot. So we're far behind with EHR data where we are now with NLP. We obviously want to do prediction, but many times just figuring out if somebody really had a heart attack is hard, let alone figuring out if they're going to have one 5 years from now. So the computable phenotyping problem is really tough. But then whenever I give a predictive model to the clinicians, they really want to look at attribution. So what was it about this person that led us to have that event? And therefore to answer the counterfactual. If this person had not been on this drug, like when Vioxx was still on the market, if this patient had not been on Vioxx, would they have still had that myocardial infarction or heart attack? And so this gets at causation, which much more broadly gets at causal inference, how large is the impact, or causal discovery, is this drug, is there a link between this drug and this event, or repurposing, which is the same problem but with happy drug outcomes instead of negative ones. And then of course discovery. We're building a new center here at Duke called Discovery AI, which is all about using protein language models to do new target discovery, but also new molecule design, but really to link that to the clinical data. And that new molecule might only work in certain patients, or it might have adverse events in some patients. If we can predict that ahead of time, then we can still take that molecule to market. I want to talk high level for a minute and then get into some technical items. The barriers to all of this, like I said, I think protein language models are just going to keep getting better. So that's a barrier, but I don't think it's a long-term barrier. Molecular dynamics, a lot of great people like Alex Tong are doing some exciting work in molecular dynamics. Confounding, talked about causation. All these are important technical problems to address. I think the biggest barrier right now, though, we're making progress on all of these. The biggest barrier is social and legal. We have all these data siloed and we can't combine them. And we know our models will be more accurate if we can bring this data together. So some of you are maybe in a position where you can work on those kinds of problems. I've certainly addressed those from the technical perspective of differential privacy or cryptography. What can we do to make people more willing to share data? But I think that doesn't get us all the way. We're going to need techniques like Nita Farhani and others are working on where we can can get society to think differently about how we share these data. But now I want to get back to the technical, and I want to review some old news, almost 20 years old now, I'm embarrassed to say, but we still have the same vision that we had then. So if you look at I know some of you have seen this slide from me before, but if we look at the far right column, the vision many of us have is that a patient will come in and will have their genome and their genetic data, maybe even epigenetic markers and other omics data, microbiome and so forth. But their clinical data C, their environmental exposures E, and as they're sitting in front of the clinician who now has more time since he has or she has an ambient scribe, the clinician can think, well, what is this patient at risk for in the next 10 years? I loved it a few years ago when I went to my doctor, and my doctor, she was able to say, look, you are at elevated risk right now of a heart attack in the next 10 years, and we've got to lower your cholesterol, and we've got to address a couple of other things. And, there was a predictive model behind all of that. Now, it maybe didn't come from deep learning. It came from the long-term Framingham heart study, but it was still the same idea. But also, maybe I have a condition today and we need to choose a treatment. And I would love for my doctor, I'd love for her to have a model that could say, well, drug A, treatment A, is likely to cause you an adverse event. And treatment B is not likely, given your particular history and genetics, to be efficacious. So let's go with treatment C. And so that's the vision we've all had for many years. And the way we used to plan to get to that vision is I used to have a note under here that says for thousands of different predictive targets, we're going to build thousands of different predictive models. Now we know again from that same old lesson from LLMs, we should build one model to predict all of this. And I know Jiaming has done incredible work with going back to a system from a decade ago now called Dr. AI, where that's kind of the vision. The approach we took in some early work to motivate some of what I'll talk about technically later today is we were working with the Marshfield Clinic up in the middle of Wisconsin. They've had an electronic health record for 80 years in Marshfield. And Marshfield's small, but they cover the upper half of the state of Wisconsin, as well as people who come up for vacation from Chicago and other places around the Midwest. So they had more than 1 million patients in their data when we worked with it. First thing we tried to predict was atrial fibrillation, which is a cardiac arrhythmia. It's the most common arrhythmia. It's one I had for a couple of days when I was 30 years old. So I was super excited to try to predict this. And the ROC curve is in dark blue here, if you can see my cursor. The area is maybe a little over 0.7, but I probably could have avoided my AFib if I'd cut down on all the Mountain Dew I was drinking while I was starting my research group, and if I'd taken an aspirin every day. So, with an intervention like that, you could be down here at 30% recall and have only a false positive rate far below zero 0.1, and that might be a reasonable intervention to do. Other things like stroke given AFib was a lot tougher to predict. So we started asking the question, how well could we predict everything? We'd built one-off models for warfarin dosing and age-related macular degeneration and breast cancer diagnosis or risk. So here we were not using deep neural networks. We were using decision trees because we could build thousands of different models on thousands of different CPUs in Morone-Livny's HT Condor cluster. And so just to give a brief description of what you're looking at here, because this is just more foundation for what we'll talk about next, this curve here on the right, if you can see my cursor, is a histogram of AUCs for 4,000 different ICD codes. And what you see is if we're predicting 1 month ahead of time, who will have this code for the very first time versus who will not in the next, a month from now, you get an AUC better than 0.8. If you predict 2 years in advance, over here in yellow, it's tougher, but it's still above 0.7. Because this was Marshfield, we could say 20 years from now, who's going to have this for the first time in 20 years? And it was still better than chance, although just barely, as you can see. One thing we learned that was really shocking was when we added genetic data. Marshfield had a personalized research medicine program and genetic data on 20,000 of the million patients. It didn't help us except in a few rare cases like macular degeneration. Well, let me back up first and say you could do a prospective trial on a different set of patients that you didn't use for training in the next year. And if you do that across all these diagnosis codes, this is the curve that you get along with your 95% confidence interval or your pessimistic and optimistic versions of this curve. So you're still predicting pretty accurately. We thought the genetics would really help us, and it just didn't. And we thought, well, maybe that's because we only have genetic data on 20,000 of these patients. Or maybe it's because we saw the same EWAS results in this data that everybody else found. We found the same genes associated with myocardial infarction or heart attack and so forth. But by the time you have all the patient's clinical history, you're probably already seeing that gene's effect in raised LDL cholesterol or raised blood pressure and so forth. So what we did was found a way you can use this tool if it's useful to you. It's publicly available, and the software has been written up in the journal Bioinformatics. But we found a way with de-identified data to build family pedigrees from the EHR data. Tatanetti found a different way to do it, so we partnered to build a software system that has both, or the combination of both, or either by itself. But basically, when we built these family pedigrees, which you can think of as in a high throughput way, doing old-fashioned genetics, if you will, if we take the same results we had, I'm sorry, some of them are in different colors, like the green was the yellow before, but if we take the same histograms we had before, and down in the mirror image here below, you add just a few genetic features or a few pedigree features, like has anybody in my family pedigree already had this event at the time we're making the prediction for me? Then you can see that across a wide range of diagnosis codes, you get a small but consistent bump in blue. And if you try to predict further in advance in green, you get a bigger increase from the pedigree information. Now, some of that admittedly may be shared environment. It's the best we can do at getting a kind of surrogate, if you will, for genetics in a high throughput way without genotyping all million of these patients. Let me stop a minute and see if anybody has questions so far, because I want to branch off now from this result in three different directions that'll all take us towards a foundation model for health. They won't get us there, but we think they're important steps towards that. Any questions so far? Well, feel free to jump in with questions as we go, but it won't surprise most of you if you've done some of this that when we looked at the models and showed the clinician the models, the clinicians immediately wanted to infer that the impactful variables for prediction were causal. But if you look at the best predictors, for example, for heart attack, one of the best predictors is beta blocker. And if you wanted to think that was causal, you'd be taking people off their beta blockers and increasing their risk. So the whole importance of confounding pops up there, that these people are on beta blockers because their doctors already know that they're at risk of a heart attack. And the system is just picking up on that information. And so most of the time, we don't just want predictors, we want causation, and we want to deal with confounding. And many of these confounders are unobserved. Most of the nice causal algorithms we have start by saying, let's assume strong sequential ignorability or no unmeasured confounders, the NUCA assumption. And that's just almost always wrong. So that's a big problem we have. So I want to talk about another line of work for a minute for getting at causation that we can learn a lesson from. Way back when Congress told the FDA that they had to do, and the pharmaceutical industry, that they had to do post-marketing surveillance, that the clinical trial wasn't enough. They had to keep following the drugs. There was a group called the Observational Medical Outcomes Partnership, or OMOP, formed that wanted to evaluate a lot of these methods. Around the same time, the Sentinel Initiative was formed. OMOP now has a successor called OHDSI or Odyssey. Some of you may be more familiar with them. But the original OMOP evaluation was a game where you say, okay, we've got these drug categories and we have these health outcomes of interest. And of course, you've got to do a lot of work just to figure out who's really on these drugs and to phenotype these outcomes. We talked about computable phenotypes. But if we take for now that's been done, then the game is to say, if a drug, if a drug's label has the condition on its label, like it's a no-brainer that warfarin might cause bleeding since it's a blood thinner, but if it's on the label, we'll take that as a ground truth adverse drug event. The task is pretending you don't know that already. How well can you rediscover it? And so a big lesson here was, I think Odyssey right now makes a lot of use of propensity scores and either inverse probability weighting or Fan-Lee's overlap weights. But the big lesson, at least in this case, was that when you try to address this task, the unmeasured confounders are the big problem. So here's just a pictorial of the rediscovery task or the OMOP evaluation. Here are 10 health outcomes of interest labeling the rows, 10 drug categories labeling the columns. If there's a red block, it means it's on the drug label, that the drug, like ACE inhibitors, can cause the event, angioedema. If it's green, it actually helps prevent the event, so we're just going to ignore those for now. Everything else, including the green, is a potential false positive for adverse drug event. Some of these false positives, though, actually have some literature evidence. So the blues are a sampling of the non-reds that don't have, or it didn't at the time, have literature evidence that they could be a causal effect of the drug. So the task is to rank drug condition pairs from most likely adverse event down to least likely. And so a big message here, just to jump ahead, is that the unobserved confounders were a big problem. So arguably the best performer was something called self-controlled case series, a CCS, which essentially perform a Poisson regression to predict risk of the event given the different drugs that a person takes. So you're just learning the coefficients on the drugs, the weights on the drugs. So it's a linear model to predict event risk given the drugs that a person's taking. So that acts by having a patient-specific baseline. So around this time, my colleague at Wisconsin, Jamie Thompson, contacted us and said, can you use this approach for for drug repurposing or for happy side effects of drugs? And we tried, and it sort of worked. And then my student Charles Kwong realized that, well, for numerical outcomes like cholesterol, blood pressure, fasting blood glucose, or A1C, for numeric outcomes, these are changing gradually over time anyway. So here's a task where we're trying to predict fasting blood glucose. And what Charles is doing here is using an ordinary least squares regression, that's all this first piece is, with a lasso penalty to find the coefficients on the different drugs in order to predict fasting blood glucose. But now we're going to say every patient not only has their own baseline, but the baseline changes with time. Well, to get anywhere there, you've got to regularize with some sort of penalty. So the penalty here is if you have two consecutive measurements like these two, within some period of time of each other, let's say 6 months. So that's this delta here, just within 6 months of each other, that's the time of these measurements for this same patient I, then the baseline better not change, or if it changes, so this is patient I, their Jth and J plus 1 measurement. If it changes, you pay that change in a penalty. And so to make a long story short, the simple reason this works is we're saying if a bunch of people have their blood glucose drop this month and they're all on the same drug, we're going to put the negative coefficient, the weight for that change, on the drug and pay this one-time lasso penalty for that coefficient. Whereas if the patients are all on different drugs, we have no choice but to say their baseline changed. And so we pay this penalty repeatedly for every patient, resulting in a much larger penalty. So when we tried to evaluate My colleagues at Marshfield gave us a diagram with all the drugs in green that lower glucose, all the drugs in red that elevate or don't change glucose. And if we didn't use a patient-specific baseline, half of what we recovered was red or wrong. If we used a patient-specific baseline shown on the right, we got rid of most of the red. And when we use the approach I just described with the time varying baseline, I'm not arguing in general it will always be perfect, but it got rid of all the red or the unobserved confounders owing to other things changing with these patients. We then took that idea back to the OMOP task, and OMOP had many different settings based on the phenotype definitions and drug exposure that I described earlier. But here's self-controlled case series in blue across all these different settings. And here's this baseline regression I just showed you, which was outperforming it. So I'm not saying this works for every task, but I am saying for a lot of real world problems, there are a lot of events that happen more than once with some frequency in patients. And using these patient specific baselines, sort of like in Cox regression, or time varying baselines, sort of like in time varying Cox, you get rid of a lot of that confounding. Up until the results I show you next, we couldn't prove that you get rid of that unobserved confounding, but we're making some real progress in proving that now. Okay, I want to go next to, I'm halfway through, I got 30 minutes left, so I want to spend 10 minutes each on three lines of work that take us from here, from prediction and now causation, towards a foundation model for health. And we're going to get more technical with the second half of the task. So most of this was applied. It's background to illustrate the problems we feel like we're facing. And then the rest of this will be more algorithmic. Any questions at all? Okay, we'll forge ahead. With my colleague at Wisconsin, Somesh Jha, but this is all work after I moved to Duke, with Somesh and Prasad Chalasani and their student Jihye Choi and Nils Palumbo, I and Matt Engelhard and some others started working on, like many of you, LLMs, and in particular LLMs with retrieval augmented generation, because one thing we wanted to do is we knew by now we were overfitting that OMOP ground truth. It was just 10 drug categories, 10 health outcomes of interest, but the literature is full of other adverse drug events. And in particular, we want to move from those linear models to deep neural network models, which are capable of uncovering complex drug-drug interactions and drug-patient interactions. So we wanted to bring those same ideas of a patient-specific baseline and a time-varying baseline to deep neural networks. But we knew we needed a much richer ground truth. Enter LLMs, which could pull the ground truth from the literature and from the drug labels for us. But of course, LLM could hallucinate and give us the wrong information. So we wanted it to just use trustworthy documents and drug labels and possibly notes in EHR data about what doctors thought was going on. So to do that, we used a RAG system called Langroid that Prasad and Somesh had built. But you know the general setup for a retrieval augmented generation system, which is we have some LLM over on the right. We have trusted information sources, maybe trusted more than just the general arbitrary internet, such as literature we trust in green and EHR data in blue. So we're going to use a local language model, in this case it was Llama 2, to generate an embedding of this trusted information to add to our human query so that we're embedding relevant information when this query goes in. And Jihye and Nils actually went a lot further. It's very popular now to have multi-agent RAG systems. And so they had an agent that was an expert in drugs and an agent that was an expert in health events and EHR data. And the drug agent in turn had a critic agent that could improve things. And with this multi-agent process, we were able in an almost unsupervised way now to also get uncertainty quantification. How sure were we of each adverse drug event? And as a result, to recover, these are two different versions shown, and the version on the right, which incorporates the uncertainty estimate, was able to almost exactly recover the OMOP ground truth, giving us trust that this would be a good evaluation technique for us as we go forward with what I'll talk about in the next two parts of the talk. Any questions at this point? Okay, so the big picture goal here was to try to bring the same idea of addressing unobserved confounding with patient-specific baselines, bring that idea from self-controlled case series and Cox regression into deep neural networks. And that might be neural point processes or recurrent neural networks or transformers, anything that can deal with the kind of longitudinal patient data that we're working with and evaluated against this LLM-based literature. Early on, we tried to bring the patient-specific baseline idea into the neural network, and it worked pretty well, but when we went to the time-varying baselines, it just failed miserably computationally. And the reason is when we had a simple linear model, we could do an ADMM-like optimization approach, an alternating approach where we train the linear model, then re-estimate those baselines I showed you, like what would my baseline fasting blood glucose be if I weren't on any drugs today? And then having re-estimated everybody's different baseline at every different time point, we could re-estimate the model and go back and forth until we converge, much like in an expectation maximization algorithm. So here was just adding that. This is neural SCCS and a variant of neural SCCS, which did outperform the multiple SCCS from the OMOP experiment and greatly outperformed the deep neural network, but still left a lot of room for improvement. So we wanted to bring patient-specific baselines into that. This motivates the next piece I talk about because we felt like we could bring these patient-specific baselines in a principled way if we could view neural networks as probabilistic graphical models, as Bayes nets or Markov nets. So I'm going to tell you now the two punchlines that I'll spend the last 20 minutes of the talk on. First punchline is I'm going to claim that any architecture neural network at all, in the simplest case, if it has all sigmoid activations, can be viewed as a Markov network. And so all the tricks that we would use when learning and interpreting a Markov network, we can use in neural nets. And secondly, is I'm going to give you a hand-wavy argument for what we're working on actively now, which is to say, if we have other activations like ReLU, for example, then layer normalization or batch normalization are just fast approximations to proper Markov network normalization. And modulo the errors in that approximation, the argument extends to all of those once we've normalized. And then the second piece that I'll move to is to give you a formal model different than the famous Rubin model of causation, rooted in pack learning theory. And I'll show you a proof about why self-controlled case series work that argues that they actually are adjusting for unmeasured confounding. It's a proof that hasn't existed before. And we're hoping, this is work in progress, but we're hoping to bring that to this neural network view and tie these together. Okay, so let me make the argument briefly to you about this claim that every neural network, if we have sigmoid activations, is a Markov random field or a Markov network. So like many of you, for years I've taught Markov nets, Bayes nets, and neural nets, and logistic regression. If we have a neural net with just one sigmoid unit, it is logistic regression. If we're doing cross entropy loss, that's the very same. Minimizing cross entropy loss is the very same as maximizing log likelihood. This is also a log linear Markov random field, where the features are just pairwise features that are only true if both x1 and y are true, or x2 and y are true, and our weight on that feature is the coefficient. What we thought would be the case is that when you go to a deep neural network with your inputs and your output and a cross entropy loss at the end, that that would directly translate, that would extend to a deep version of this Markov random field with lots and lots of layers, where again the features are just features true if and only if the node on either side of the edge, the nodes on both sides of the edge are true. And the weight that we've learned in this neural network is the log weight in this Markov random field, just standard log linear Markov random field. This would all of a sudden give us not that the neural network is a machine for computing the probability of y given x, but it actually is a joint distribution over y, x, and all the mysterious hidden variables in the middle, giving us a proper statistical understanding of the entire neural network. But you know how research goes. Sadly, it turned out we were wrong for two reasons, and I'll show you those two reasons right now. When we train a neural network, we could view it as having a different neural network for every example, or every patient, if you like, but having the weights tied, as I'm showing you here. If I change that so that how do I want to say this? Every node has its own copy, its own unique copy of all of the ancestors, but the parameters are shared across all the copies, as shown here, then that gets us partway to the result we want. The result we want is to have a Markov random field where the gradient, because you learn Markov random fields by gradient descent, is the very same as the neural network gradient. That's what we're after. That gets us partway there if we reason by variable elimination, but it doesn't quite get us all the way there. So let me jump ahead just a little bit further. Let's see. Okay, I'm missing one key slide that I wanted. Okay, I'll just talk around it. I'm sorry. I had shortened this and I'm missing one key slide that I wanted. So the important point here is that this node and this node both have the same parents. So if I go back to here, this node and this node both depend on the same network that goes beforehand. So all I've done here is give this node and this node entirely independent copies, but with the weights all shared. The other piece that I have to do now is to say that each time there's an edge, we actually have k copies of this edge with the weight divided by k. And as k goes to infinity, so obviously we're not going to build this, it's just a way of understanding the neural network. As k goes to infinity, the gradients become identical. And so the neural network looks just like the Markov net, or here because the arrows are directed, the Bayes net. It looks just like the Markov net or the Bayes net, except the neural network is making a big simplifying assumption. It says this node depends on both of these nodes, and they have well-defined probability distributions given the input. This node depends on those same distributions, but the value of this node doesn't have to be tied as it influences this and this. So in other words, this represents a distribution that influences these two nodes, but the values don't have to be tied. That's what we capture by giving them their own copy. And then what it turns out, to make a long story short in the proof, the infinite copies of infinitely small divisions of this weight, what this basically avoids is backdoor pathways of information flow in the Bayes net or Markov net. So if you make those changes, then in fact you get the result that the neural net and the conditional random field or the Markov random field have the very same gradient. And therefore, we can take that Markov random field or conditional random field as the meaning, as the proper meaning of this neural network. And we don't have to just think of the hidden nodes as machinery to compute the output. We have a proper joint distribution over X, Y, and H, all the hidden variables. So that's the argument. Anybody want to ask a question about that? Let's see, do I have the slide here? Okay. This was in NeurIPS last year, so I thought I had the link to it, but I don't. The co-first authors were Boyal Lee and Alex Thompson. So please do have a look at that if you're interested. But why does this matter? The reason it matters is that when we're training a Bayes net or a Markov net with lots of hidden variables, we assume some prior. But in neural nets, we don't really do that. We just build an over-parameterized model and randomly initialize. And I would argue that what we're really doing is building a huge number of possible priors in by having that over-parameterized network and randomly initializing. And somewhere in there are the right priors. But if we really have information, if we have real meaningful prior information, I would argue that we should do what we do in the Markov net, Bayes net learning case. So here's a concrete example. Imagine we have some observed variables like myocardial infarction and Vioxx, which I talked about earlier. Vioxx doubled the risk of MI and hence got pulled from the market. And we have, do I have a hypertension diagnosis? Do I have a hypercholesterolemia diagnosis? Or what's my blood pressure? What's my LDL cholesterol level? And we have some embedding. I've just given meaningful words to these embedding variables like lung health and heart health, but we have some embedding here. But what if I want to say, also, I think every patient has some hidden risk for an MI and a hidden propensity to get prescribed Vioxx. If you like, I could include arrows to all the observed features. So this is almost like another embedding variable. But the one thing I'm going to do, the two things I'm going to do differently from this new variable is I'm going to put a really big positive prior weight here. It's not going to be a small non-zero weight. I'm going to put in a big prior weight like 10 here and big prior weights here to say if this baseline risk is high this month, it'll be high next month. And if this baseline risk is high, then there's a good chance that I'm going to have an MI. The second thing that I'm going to do differently here is I'm going to untie the bias term right here, just one parameter in this whole big neural network. I'm going to untie this across the patients. So each patient will have their own bias term, but it's untied across the patients. This becomes my patient-specific baseline, and it can change over time, but in this controlled, smooth, weighted fashion. And so that idea comes entirely from viewing the neural network as a probabilistic graphical model. Now, we're still very much in the process of doing the experiments with this approach. There are a lot of interesting questions like, should this be a vector? Should this become part of the embedding and we just change this bias and this weight? Everything else is just like another embedding variable. And then should this be a small vector instead of a single value? And this is still very much work in progress. But while the empirical work progresses, we also want to try to do a proof about this. So propensity scores, inverse probability weighting, has really nice proofs. But even self-controlled case theory does not have a causal proof associated with it. It's just viewed as an estimation technique that works well in practice, but it didn't have a causal proof. So I'm going to switch gears now to this idea of trying to get a causal proof. And the hope is that it can help us come up with a good combination of these different approaches that people see are useful now in causal reasoning. Causal transformers get a lot of attention right now, but they only address observed confounders. And as we saw in the OMOP experiment, it's the unobserved confounders that are the issue. So I would claim we need new methods that are at the intersection of these approaches, and we need a new model that will let us prove positive results for techniques that work well in practice, like self-controlled case series. So I'm going to pitch that model to you in the remaining time that I have left. Back when I was a grad student at Illinois, I was doing a lot of stuff with pack learning. And now we're all in a tizzy about, well, over-parameterized models have really high VC dimensions. So how does this relate to pack learning? And people are making good progress on sorting all that out. But I just want to review the pack definition briefly for you. For pack learning, we say that a concept class C, like decision trees, is pack learnable, or a concept class over a possible target concepts like decision trees or depth 2 decision trees defined over a space of instances like n-dimensional feature vectors. We're going to say that a learning algorithm can pack learn that concept, say using a different hypothesis space H, or otherwise it's implied to be L, if for all concepts in C, so this is worst case, all probability distributions over X, and any epsilon and delta that the user specifies, with probability at least 1 minus delta with high confidence, the algorithm is going to output a hypothesis that has low error less than epsilon. And the more complicated the concept, so the size of C, or the instance space, or the smaller epsilon or delta, the more time and examples we get. We want to play the same game with causal discovery. We want to say that when SCCS addresses the OMOP task, we can't expect it to be perfect. But if you give it more and more data and time, then it can get better and better. That's kind of what we're after. So you can go see this idea on arXiv. It's pack discovery, but PACC, or pack causal, or PA with C with one C, causal discovery. And the idea here, I want to give you the intuition in the time we have left. The learning algorithm is going to see a data set drawn entirely from one causal model, but we're not sure which one it is. And for reasons I can get into about the intuition, we're going to treat this like like hypothesis testing. And we're going to, in hypothesis testing, we have a null and an alternative hypothesis. So here we're going to say there are two possible causal models. We're not sure which one generated the data, but if we get enough data, we'll be able with high accuracy to say which one generated the data. So the models are arbitrary probability distributions. They might be Turing machines or something else. We're going to say that the problem is pack discoverable if for any epsilon, given sufficient time and training data with high probability, we're going to correctly identify which model generated the data. You can scale this up to find the right model among many possible models by, instead of having one pair, having many possible pairs. And so the formal definition do that. You have a family of pairs. And I'll just give one concrete example. Suppose our probability distributions are represented by Bayes nets, and we want to know, is drug A having a causal impact on event B, like Vioxx and MI? Is it having that impact? Is there an edge here, or is there not? So these are the two models. Of course, to have complete models, you have to also put your probability distributions, your conditional probability distributions in there. We handle this by having a family of possible pairs of models, all with these same two structures, but the probabilities can be different. And then we have to ensure with a parameter delta that if there's an edge from A to B, that the influence is big enough, right? You could have a distribution that's representable by either of these. And so we've got to make sure if the edge is required that it has some kind of impact. And so that's the task, is to distinguish these. And by having a family of pairs like this, we're finding all possible pairs where there is an effect from A to B versus not. And so that defines this concept of an edge from A to B. And that's the task. So with SCCS, and it turns out, based on Bayes net results, that you can prove a positive result there. But more interesting, for self-controlled case series, you have a drug D, an event E, and possibly ridiculous numbers of unobserved confounders. You can be a whole big vector. And what we're able to show is that if the unobserved confounders can't change with time, then SCCS PAC discovers whether or not there's an edge from D to E. And so pictorially, the assumptions look like this. Sorry, I've switched from D and E to A and B now in this picture. I apologize. But the picture looks like this, and if you can't change with time, then we show that in fact SCCS succeeds. But what we really want, as you remember from the earlier motivating parts of my talk, is we want you to be able to change with time. If it can change in a way that's unrestricted, the problem's impossible. An adversary can change you in such a way to make this just impossible. But if you can place some limits, some regularizers on our assumptions around how you can change, then we hope that we can get a positive PAC result for the neural net approach that I described before. Unfortunately, that will have to wait for when you invite me back, I hope a year or two from now. So I'm going to end with that open question. Is there a way we can let you change with time and still get a positive result? And our neural net's a good way to get that. So with that, I'll stop there and take any questions. Thank you. Thank you so much.
Thanks. Thanks, David, for the wonderful talk. I mean, this is really, I think audience will benefit tremendously. You put in a lot of the new work that you published towards the end. That's very, very interesting. So for the audience, if you have a question you want to ask Professor Page, please just put it into the chat.
And while we wait for that, I do want to say maybe I don't have a conclusion slide, but if I had a conclusion slide, what I would say is if we can find a way to make transformers or some other deep neural net address these unobserved confounders, then I think that's the architecture we should be using for a foundation model for health.
So maybe I can start with some specific questions, then I have some general question also. I don't know, we only have a few minutes. I don't know if you have another meeting in 3 minutes or you can stay a little bit longer.
No, I can stay longer. I'm sorry I ran over time, but I can stay
Oh,
longer.
no problem. So that would be great. So on this last piece this is very interesting. This, I guess, this new causal framework connect pack learning for this causal.
For causal discovery, yeah. Yeah.
And so how does this new framework changes because the traditional process?
That's a great question. So we do have some results in the paper that show that anything you could prove in the Rubin model. So right, so we're always working with this conditional outcomes framework, and we have some really elegant notation, and we can prove that inverse probability of weighting achieves certain properties if there are no unobserved confounders. A lot of those results are saying we're guaranteed to make the right causal discovery or the right causal inference under these assumptions. All those results can be cast in this PAC discovery framework, but also all the results in the Pearl style approach or Robin style approach with graphical models, all those results too, like Pearl's do calculus, can be cast in this framework too. So our argument is that this framework generalizes those, but the importance of the generalization is that now you can prove some new results you couldn't before. So a lot of the proofs, rely on saying if you assume no unmeasured confounders and two or three other assumptions, you're guaranteed to make the right causal inferences. But that's too strong of a claim to make for something like self-controlled case series, which is why it had no theorems and proofs like that before. And so now this setting kind of does for causal discovery, we think what pack learning did for identification in the limit. It says you don't have to get the right answer at any point. You just have to, with high probability, get a sort of good answer. And the more time and data you have, the higher the probability and the bigger the constraint on what sort of good means. And we're arguing that this lets you express all the theorems you could before, but more theorems now too.
It's very interesting. Just a quick follow up on that. So in the real world situation, right? So you have very high dimensional data. So when we move into this high dimensional settings like EHR genomics, so what are the main bottleneck? I mean, is
Yeah, yeah.
frameworks do like the old ones that has those type of limitation, or now?
Yeah, so it's still the case that when you move to high dimensional data, you're polynomial in a much bigger number, so you do get more time. So we want that to be a very low order polynomial for it to be practical. Actually, the biggest bottleneck we have is the following. Yeah, I argued that all neural networks can be viewed as infinite infinite width tree structured probabilistic graphical model. Tree structured PGMs are nice because you can prove pack results for them. So if you let me for now replace these infinite width tree structured PGMs with just simple tree structured PGMs, then we can prove some of these results we want right now. The big problem is the gap that still exists between neural nets and pack learning, that they're working well in cases where we don't have a pack result still.
Mhm.
And so that's going to be our gap for proving theorems in this world too. So our approach for now is going to be at first just to replace the neural network with a tree structured PGM and prove the theorem we want, and then show empirically that it works well with the deep neural networks.
Interesting.
But
Fast.
the theorem we want to prove most is really specifically for this model or a model that looks like this but has a transformer instead of an RNN. And so we're just adding this extra linear sequence of embedding variables that get their own baseline at the start, different for each patient.
Yeah, interesting. Very interesting. So I have some general question, but I saw there's audience questions
Okay.
in the chat. So if you,
Let's see. Let me see if I can see if I can see the chat if I stop sharing. Should I do that, or do you want to read them out to me?
I
You can speak if you want.
yeah, yeah, you can ask the question. Yeah.
Yeah, sure. So the first question is, do you think the large language model can use common sense knowledge or after the fine-tuning the domain-specific knowledge learned to infer the observed confounders, like to do some discovery works?
Oh,
Because
that's
you
a great
also
question.
mentioned that you can use the large language model as a help to do some RAG and to do some grabbing of the information. And the second question is, would a graph neural network be a better like structure compared with the transformers for modeling the causal relationships, since the event order and the direct or indirect links are matter, and also the Bayesian network, maybe the graph neural network can better reflect the relationships compared of the existing transformers used by the most of the large language models.
Yeah. Okay, that's a great question. Let me start with the second one. That may very well be the case. We're also interested in graph neural networks. The transformers are taking sequential data, even though they're using attention instead of the directed arcs. And so we are interested in asking, can you replace causal transformers, which assume only observed confounders, with something that can handle unobserved confounders? But you're right. At the end of the day, we might find that to get the theorems we want or to get the empirical performance we want, we are going to need to move to something like a graph neural network or maybe go back to either RNNs or neural point processes, which are things we've long been interested in as well. So I think the graph neural networks are a big interesting item there too. On the first question, this is a great point. So there is some exciting work recently on trying to find surrogates for unobserved. So the problem is if the unobserved confounder really is not just unmeasured, but we haven't even thought of it, which happens sometimes in the world, then I'm putting this patient-specific baseline in as kind of a placeholder for whatever that might be. But certainly, if we could find surrogates, if we could compose measured variables to create some model, if you will, of this unknown, unmeasured confounder, that would be, I think that would be fascinating for two reasons. It could give us better performance and could give us insight into what this unobserved confounder actually is, what it means. And there is interesting work trying to find surrogates that you build out of observed variables for either the confounders that are influencing the treatment decision or the outcome. And can you take that even further and more fully characterize these confounders? I think that's extremely interesting.
Cool.
Thank you so much. I used to think about some way, like maybe the large language model can help generate something, whether true or not. But yeah, it's hard to verify if that the discovered variable is actually that the confounder we desired or not. So I think that's a bottleneck I face too when I think about this.
That's a great point. I like that a lot. And maybe there are ways we can evaluate that. You know, the hard thing with evaluating causation as compared to prediction is prediction, we can just hold out some data or do cross-validation. But for causal discovery, we really have to work on rediscovery problems. So maybe there are ways we could do that where we pretend we don't know something and ask, do we recover it? Unfortunately, that's maybe an easier problem for the LLMs because if we're pretending we don't know something, it's still in the literature and the LLM will see it. So that
Yeah, that's true.
evaluation is tricky. You're right for that.
Thank you so much.
Thanks for the question. David, I have a follow-up question on the validations of those models, right? Today, the success of all those foundation model is it can support very many tasks, right? So tasks that you never really designed for that can give a lot of times correct or reasonable answers, right? So in this case, those standard machine learning metrics like accuracy for a certain task are no longer probably sufficient. Have you thought about what's the best practice today for validation?
Yeah, you know what I've been doing? I've been punting that question over to Michael Pincina and Nicoletta Economo, who are here at Duke, and they've for years run something called ABCDS testing, algorithm-based decision support for all of Duke Health. And so once we started getting ambient scribe software and all these other things, they took that problem on. How do we evaluate these systems that are generative AI or aren't just selecting from predefined possibilities? And they've been trying to expand what they're doing to a larger coalition that you might be part of called Coalition for Health AI or CHAI. But to be honest, I've I used to do a lot of work on evaluation and I don't know how to evaluate that. So I've been relying on their work and others to try to solve this for us. We've been in a lot of conversations with Somesh Jha. How do we evaluate this stuff? And I just don't have a good way. I don't know if you're working on that
Yeah,
problem
I
right.
mean, everybody has to do some. These days, evaluation seems to become the creative, cutting edge things.
Yeah, it really is. It really is. And it's
Question maybe, but you mentioned this multi-model medical data, right? And so if you're trying to build this foundation model on this multi-model clinical or like a patient type of data, do you have some kind of intuition? What modalities would you prioritize first as like taking note?
Yeah, this is just me because of what I've worked on, but I would prioritize the EHR and the medical image data. You know, the medical image data is much more objective compared to the EHR, which can get coded in many different ways depending on the doctor and their constraints. So I'd prioritize, I think, EHR, including notes, but also the image data. And then maybe this is again just more me, but I'd go to the other extreme and instead of I'm very fascinated with mobile health data, but I'd go to the other extreme and say, with protein language models and their use now to try to generate new small molecule structures or ligands, can we right now bring that idea of PLMs and molecular data together with the EHR data and do better than we could do with just the EHR data alone at some interesting problems like drug repurposing or adverse drug events, for example. Maybe don't go all the way to
Mhm.
inventing a new drug just yet, but asking, can that molecular level data help us do a better job than just with EHR data alone of finding a new use for an existing drug,
Mm.
for example?
Interesting. Yeah, I think maybe I'll ask you finally this very high level advice type of question for our students and early stage career researchers. So maybe a general advice, what would you give to the students who want to pursue research in this AI, healthcare space? And also today, I think that was not the problem maybe 5, 10 years ago in terms of compute and data set, right? I guess data set is always a problem. This computational constraint were not so much, right, in the research.
I'm battling all these problems, so I'm just going to be thinking out loud here
Mm.
and looking for a solution with you rather than giving a finished solution. I often, even before the dramatic rise in LLMs, I would often say that the magic happens when I can bring together the really smart people, the PhD students, and the data and the compute resources, like we did in that high throughput
Phenotyping.
stuff we talked about earlier. And that's always that took us 10 years just to solve that practical problem. And that was with data from one site. And then we really want to bring data together from multiple sites. And so that's always been a really hard problem. But now the game has just gotten scaled up orders of magnitude around the compute power with the GPUs and the need to maybe just bringing the medical data together with a foundation model already trained on all the GPUs is hard. So this a fourth thing that comes into the mix is the foundation, the existing foundation models that we want to use that aren't open source or open weight. So that's four things that are really tough to bring together. And so there's kind of advice for right now doing a PhD and advice for career. And my advice for right now doing a PhD is try to do an internship at one of these big companies. But if you're constrained around compute power, around data, then think about what work we can do with what we have. So for example, if somebody doesn't have the data and compute power they need, they might decide to think about the next step of proving some theorems in one of these frameworks that I described, either the neural nets as PGMs or the, excuse me, the pack causal discovery stuff. So some people may want to, for now, take a more theoretical approach. Or if they're working on causal discovery and they can't get all the data they need, but they have the compute power, then maybe work with synthetic data or be willing to do some things with big compute with synthetic data and then some different things for a while with the real world data. And so
Mhm.
we do have to work around the real world constraints that we have. But then I worry big picture, career long, is it the case that all the real action is going to happen in the future in industry because that's where the compute power is? So maybe those PhD students do the theoretical work now and then they need to go off to a big company that gives them the compute power. I hope that's not the future. I hope that those of us at universities can find better ways to partner with industry. But right now, with things going on in the federal government, universities are worried that if they have somebody working jointly for industry and the university and doing grant proposals to the federal government, that they might get in trouble. And so, I hope that the federal government and the universities that we can find a way that our students, for example, can have joint appoint or our faculty can have joint appointments in companies, get the access to the compute power on the one hand and the access to the data and the biologist over at the university on the other hand and bring that together. But I think that's a problem that we as faculty and the administrators have to solve. And for the students, my advice is to take a good cold hard look at what your constraints are where you are and what can you do subject to those constraints. And then in planning your future career, think which there are constraints everywhere, but think which set of constraints do I want? Do I want to be in a company where I have lots of compute power, but maybe universities or health systems aren't willing to share the data with me? Or do I want to be in a health system where I get the EHR data, but maybe I don't have the compute power? And I hope we as senior leaders can solve this for the students. But until we do, I think the students are going to have to make decisions about what their constraints are, how they work now, and then what their career trajectory might look like dealing with those constraints. I'm sorry I don't have a more optimistic answer, and I know that was a long word, a wordy answer, but some of this is on us